So, thanks for your kind introduction. I'm Ruyi Chen. I'm PhD student at the University of Queensland. And today I'm going to present my recent work about using the multimodality model to improve the zero-shot prediction of the disease effect on proteins. The first question, how to predict the mutation effect on proteins? And this is the answer. A lot of teams train different models and using the different features based on different algorithms. They are from collect this information from the protein sequences, structures, and homology-based features. And we want to know if there are any other source of information can be further used to improve the disease effect predictions. So we got some idea from the natural language processing. Just as the English and the Germans, they may have the similar, they may can convey some similar meanings, but have some minor difference. I think some of you may know what is this German words represent, but I can't understand. I just used some sentence from the books. So the genetic informations just also have this kind of translation, start from DNA, mRNA to proteins. Our hypothesis is the languages model training based on the same regions, the coding regions, but expressed in different languages, such as the cDNA and the proteins. We may also have these linguistic characteristics. So if this is true, we can combine the two languages model together to improve these disease effect predictions. So we collect, for our experiment design, we collect the missense mutation from the ClinVar database, and these missense variants can be further divided into likely pathogenic, benign, and benign. And then we choose these high-quality missense variants, and then with at least one review start. And we use the two different languages model. One, they are all transformer-based and training with the mask language modeling objective. The first one called the COM. This is a codon languages model that it took the wild type cDNA sequences as the input, and it tokenized these cDNA sequences into 64 different codons. And then the second one is ESM2, a protein languages model. It takes protein sequence as input and tokenizes the residues into 20 different residues. And then we calculate the log likelihood ratio generated by the two different models. So our hybrid model is built upon when we calculate the two effect score calculated by the different models. We use the Pearsman optimization to identify the coefficient of the both terms. And finally, we find if we assign the equal weight to these terms, they can always get the best result. So this is our result. We find our hybrid model achieved a 3% increase in the ROC AUC in classifying the disease effect, and it significantly outperformed the two single sequence-based languages models. But the next step, we want to know why we combine the PLM with CLM can improve the disease effects predictions. So we calculate the differences between the two effect score calculated by the PLM and the CLM, and then they are falling at the rejection regions. And then we further categorize this mutation into codon languages model better and the protein languages model better. Codon languages model better means it will assign a more negative value to the pathogenic variant and the higher value for the benign variants, so that the model can distinguish them efficiently. So and the next step, we define the bias ratio, and it is a subset variant pair frequency divided by the overall missense variant pair frequency. And if it's value more than 1, it means this mutation is enriched, otherwise it's not enriched. And finally, we conduct the chi-square test to access the statistical significance. And then the color in this plot means if they are red, they are impressed, and if it's represented by a star, means they are significantly enriched. So we find the primary differences between the PLM and the CLM effect score lies in these mutations changes from the amino acid with a high codon to the low codons. And then for those mutations that do not substantially change the codon usage, the effect score assigned by CLM and PLM are generally similar. The second thing is we find the PLM and CLM differ in prediction the specific mutation types, such as the CLM is better at prediction the missense mutation from T to M, while the PLM is better at prediction the reverse. So PLM and CLM are complementary. So thanks for your attention. Thank you.
Thank you very much. Very interesting work, Ruyi. So we'll go to the next speaker for this session.